Hello my dear aspirants, I hope all of you are doing well and I hope your preparations are going really well because we don't have much time left for your Northeast SLET exam. So I have with me some important topics which are really important for your SLET exam. So watch the video till end and these topics will be a good revision for you. If you have any kind of doubts, you can comment down below. I will be again back with this type of topics till your exams. We will do proper revision. So watch the video till end, and if you find my videos helpful enough, you kindly share these videos with your friends also so that they also get benefited. And yes, if you want to take detailed coaching of Northeast SLET from me, then you can comment down below or you can contact in the given number. The first topic that we will start for this revision series is your GPCR, and this particular topic is from your unit cell signaling, and this is one of the most important topic from the cell signaling unit. So we'll not discuss everything about GPCR. I hope you already know. We'll just see the most important pathway of GPCR, which is your CAMP pathway, CAMP pathway. So before starting the pathway, let me quickly tell you some common and important things about GPCR. GPCR is a membrane bound receptor or a membrane bound integral protein which can pass the plasma membrane 7 times. That's why it is called serpentine receptor. In the cytosolic part of the receptor, one G protein is attached. G protein is such a protein that has a GTPase activity that can switch between GTP and GDP. This particular G protein is made up of 3 subunits, alpha, beta and gamma. When there is no ligand, then the alpha subunit is bound to beta and gamma subunit. Also, there will be GDP bound to the alpha subunit when there is no ligand, and when there will be ligand, the receptor will be active, and instead of GDP, GTP will bind to the alpha subunit, and alpha subunit will detach from beta and gamma, and alpha subunit will continue the process. So with the help of CAMP pathway, let us understand this better. So the main function of the CAMP pathway is glycogen breakdown. Okay, so glycogen breakdown is the function. Apart from this, there are many properties of CAMP pathway. Anyway, let me draw the receptor first. So this is the GPCR. Ligand will come. Ligand will bind to the GPCR. GPCR will get activated. Active GPCR can act like GEF. Now this GEF, what is GEF? GEF are such enzymes or proteins that can convert GDP into GTP. Now this active GPCR will act like a GEF, and here alpha, beta and gamma, this entire G protein will be now active because this GEF or the receptor will now add GTP to the alpha subunit. As a result, the alpha subunit will get detached from beta and gamma subunit. This alpha subunit will activate something called AC, adenyl cyclase. This AC can make CAMP, okay, cyclic adenine monophosphate. This CAMP will activate one protein called pKa. Now if you zoom the pKa looks like this. It will have 4 subunits, okay, 2 regulatory subunits and 2 catalytic subunits. CAMP will go and bind to the regulatory subunits, 4 CAMP. Total 4 CAMPs will go and bind to the regulatory subunit, 2 CAMP each on one regulatory subunit. As a result, what will happen? The catalytic subunits will get detached from the regulatory subunits. These catalytic subunits, what it will do? It will enter the nucleus. Inside the nucleus, what the catalytic subunits will do? The catalytic subunits will activate the transcription factor or a protein called CRAB. Okay. Upon activation, the CRAB will go and bind to the DNA, to a specific region of DNA, specific gene, gene X for example. This gene will get transcribed, RNA will be formed, and these RNA will make a protein, protein X for example. This protein will be responsible for glycogen breakdown. Okay, so this is the objective of CAMP pathway. I hope you have understood this. And if there is no ligand, then what will happen? If there is no ligand, I am explaining it here. If there is no ligand, then the receptor, GPCR receptor, will be inactive. When it will be inactive, then the alpha, beta and gamma will remain together. The entire G protein will remain together, and GDP will bind to the alpha subunit, not GTP. Okay, in inactive state, GDP will bind to the alpha subunit. As a result, when alpha subunit is bound to GDP, that means alpha subunit is inactive. Inactive alpha subunit cannot proceed further. It cannot make AC, it cannot make CAMP, and nothing will happen. As a result, the signaling will be turned off if there is no ligand. If there is ligand, then GTP and everything will be active. Next important topic from the same unit is your Ras-MAP kinase pathway. Pathway is different from the previous one because the receptor which will be associated is your RTK, that means receptor tyrosine kinase. Okay, now let's see the pathway. This receptor tyrosine kinase is a type of enzyme linked receptor, and unlike GPCR, enzyme linked receptors can pass the plasma membrane only once, not 7 times like GPCR. It will pass the plasma membrane only once. So here you have to know the objective of this pathway is cell growth. Okay, so imagine one ligand will come. This ligand is a growth signal, for example. Then what will happen? This receptor will become dimer. Okay, in inactive form, the receptor will become will remain as monomer. When ligand will come, the receptor will become dimer, and thus the receptors will be now active. How it will be get activated? All the tyrosine amino acids, because receptors are protein, we know it well. The cytosolic part, this part of the receptor which is facing the which is in the cytosol, the cytosolic part will act like an enzyme. Okay, the cytosolic part will act like kinase enzyme. The cytosolic part of the receptor will act like kinase enzyme, and we know kinase enzyme can add phosphate in particular amino acids. So the kinase enzyme, that means this part of the receptor, will now add phosphate. Where? To its own body. Okay, phosphate to the own tyrosine amino acids, to its own tyrosine amino acids. Now this site is called phosphorylated site of the receptor. Here one adapter protein called GRB2 will bind. GRB2 will activate another protein called SOS. This SOS is a GEF. Like I told you before also, GEF are proteins or enzymes that can convert GDP into GTP. This SOS will add GTP on one protein called Ras protein. Ras, what is Ras? Ras is another type of G protein. Just like we had studied before also, G proteins are proteins that has GTP's activity, that can switch between GTP and GDP. Ras is also same type of a G protein. So previously, in the absence of ligand, GDP was bound to Ras, but now we have ligand, so SOS will be active, and active SOS can add GTP on Ras. In case of GPCR, the receptor itself was doing this particular activity, but here enzyme-like receptors will not have that property. That's why they need to hire SOS. Ras will be active. Now activated Ras will activate another protein called RAF. Okay, RAF, another name of RAF is MAP kinase. Okay, MAP kinase. RAF will activate another protein called MAC. MAP, another name of MAC is MAP kinase. MAC will activate another protein called ERK. Another name of ERK is MAP kinase. MAP kinase. ERK, upon activation, will enter the nucleus. Inside the nucleus, we have our transcription factor, phosgene. Okay. ERK will go and activate 1st June. 1st June upon activation will sit upon DNA on a special region of DNA. Because it is transcription factor, so it will transcribe the DNA. RNA will be made. This RNA will become protein, and this protein will be nothing but a special type of growth factor that can do cell growth. So this was the objective of Ras-MAP kinase pathway. Next important portion is your replication. Okay, unit molecular biology. So here we'll discuss all the proteins involved in replication. So the first protein or the first thing is your initiator protein. Okay, initiator protein. If we talk about replication in prokaryotes, first protein that will participate is your initiator protein. In case of prokaryotes, we call it DNA A. Okay, after initiator protein, who will come? Helicase loader will come. Helicase loader in prokaryotes, we call it DNA B. After DNA C. After that, who will come? Helicase. Helicase in prokaryotes, we call it DNA B. After that, who will come? Your primase enzyme. Okay, primase enzyme, we call it DNA G. Then who will come? Your topoisomerase. Topoisomerase, we call it topoisomerase 1 and gyrase. Okay, topoisomerase 1 for single strand, gyrase for double strand. Then who will come? Your main hero for replication, which is your DNA polymerase 3. Okay, DNA polymerase 3, we call it DNA polymerase 3 only in prokaryotes, the main replicative enzyme. Then we have our ligase enzyme. Okay, we have ligase enzyme. We have SSB proteins also, single strand binding protein. And in eukaryotes, all the names are different. Okay, these are for prokaryotes. For eukaryotes, the names are different. The initiator protein in eukaryotes is called ORC. Helicase loader in eukaryotes is called CDC6 and CDT1. Helicase in eukaryotes is called MCM227. Primase in eukaryotes is called DNA polymerase alpha. Okay, isomerase in eukaryotes is called topoisomerase 1 and topoisomerase 2. Topoisomerase 1 for single strand, 2 for double strand. DNA polymerase 3, which is the main replicative enzyme in eukaryotes, we call it DNA polymerase epsilon for the leading strand and DNA polymerase delta for the lagging strand. 2 are there. Okay, here only one, DNA polymerase 3 is the main one. Here 2 different polymerases are there for the leading and lagging strand. Others, these are same only. And this SSB protein in eukaryotes is called RPA. So you need to remember all the names of the enzymes. In prokaryotes is different, in eukaryotes it's different. Discussing replication in prokaryotes. Eukaryotic replication is almost same, but there are some additional steps. Anyway, let's discuss prokaryotic only here today. So at first, what will happen? Suppose this is the initiator protein, which I told you, DNA A. Okay, our DNA will wrap around this protein. Okay, and thus replication will start. That means initiator protein can start the replication process. After that, helicase enzyme. What helicase enzyme will do? We know there are hydrogen bonds in between our bases of DNA. So helicase enzyme can break the hydrogen bonds wherever replication is needed. Helicase enzyme will break it and replication will start at a special point which is called ORI or IC. Okay, this particular region of the DNA is rich in adenine and thymine, because we know adenine and thymine will have 2 hydrogen bonds, and guanine and cytosine will have 3 hydrogen bonds in between. So 2 hydrogen bonds are easier to break than 3 hydrogen bonds. That's why ORI-C is rich in adenine and thymine, and helicase enzyme will work here. Okay, after initiator protein, actually helicase will not start the work. Helicase loader, which I already told you, DNA C, helicase loader will load the helicase. Okay, helicase loader will load the helicase upon this region, and then helicase will break the hydrogen bonds and make a bubble. This particular thing is called DNA bubble. Okay, now if we cut the bubble in between, we'll get a structure like this, which we call replicating fork. So while studying replication process, we will use the replication fork instead of bubble because this is easier to study. Now let's start the process. I have marked the terminals 3' complementary 5', 5' complementary 3'. Now what will happen? Helicase, helicase loader and initiator protein. All of them had already done their activity. Now we'll start from primase enzyme. Okay, primase enzyme will start the work. So primase enzyme will make a short RNA primer here also, here also. So this we can mark 5', 3' and 3', 5'. After primase enzyme, who will come? Our DNA polymerase enzyme will come here also, DNA polymerase here also. But DNA polymerase can only work here because we know DNA polymerase enzyme needs special things. It will need a 3' OH end, okay, and also it will need one template. So here DNA polymerase will get a 3' end, 3' OH end, and also it is getting a template. So DNA polymerase enzyme will beautifully continue the strand till the end. Okay, but here there will be a problem. What is the problem? Here we don't have 3' end. Here the polymerase is getting the template, no problem, but there is no free 3' end. Here we have only 5' end. So the DNA polymerase will stop for a while. It cannot continue the strand. So who will come? Again primase enzyme will come. So again primase enzyme will start the work. Okay, so the primase enzyme will again make a short RNA primer. So the primase enzyme will do the work from the 5' to 3' end. It will make a short RNA primer. So here 3' end will be there, right? So another RNA polymerase will come and continue the strand like this. Again this will continue till the end. Again RNA primer will come and start the work. A short RNA primase will come and make a short RNA primer. Again third DNA polymerase will continue the strand. So this will continue. Primase will again make a short RNA primer, and polymerase, another polymerase, will continue the strand. Like this till the end we will get a DNA RNA hybrid. Okay, these fragments are called Okazaki fragments. These DNA RNA hybrid fragments are called Okazaki fragments. Okazaki fragments. This continuous strand will be called leading strand. This continuous strand will be called lagging strand. The main reason of this continuous strand is the absence of 3' end. So if polymerase enzyme will not get 3' end, instead of 3' end it is getting 5' end. That's why it will stop and it cannot continue. Again it will require primase to make short segments so that it will get 3' ends and it can continue the strand. Okay, so these DNA RNA hybrids are removed by certain enzymes like RNase H, okay, FENI. Remember, these are certain enzymes that can remove the DNA RNA hybrids because we don't need hybrids at the end, we need pure DNA. So these are certain enzymes that can remove DNA RNA hybrids.